All right, good afternoon, everybody. Welcome to my presentation. My name is Grace Novak, and today we will be reviewing RT-qPCR testing. So let's get down to the basics of DNA and RNA first before we jump into what exactly that testing is. So DNA is a double helix structure made up of repeating units called nucleotides. It contains adenine, guanine, cytosine, and thymine. The DNA holds genetic instructions and it is a requirement for all living organisms. Its primary function is to replicate information to pass on to cells in order to synthesize proteins. But in order to synthesize proteins, it needs its messenger RNA, which is ribonucleic acid, which is a single-stranded structure made up of adenine, guanine, cytosine, and uracil, where it then is a messenger from DNA and carries information to ribosomes, and then carries out protein synthesis, and regulates genes. By doing this, it is able to turn genes on and off as needed. So, how do DNA and RNA work together? So, in conjunction, DNA works with RNA to create proteins, as I said earlier, but it does this through a process of transcription and translation. Transcription occurs when a DNA sequence is copied into a complementary RNA sequence by RNA polymerase. RNA polymerase is an enzyme that goes through initiation, elongation, and termination. So at the beginning of the sequence, the polymerase will attach here, will go down through elongation, and then eventually reach the end, and which is where we see termination, which creates our mRNA. So from here, our messenger RNA goes through translation, which is the decoding of the instructions here that we've had, which will then carry out to create this protein, translating each codon into a specific amino acid, as you can see here. So, now that we have our background a little bit on DNA, RNA, and protein synthesis, what is RT-qPCR testing? So, it is a mouthful. It's reverse transcription quantitative polymerase chain reaction. You're probably familiar with PCR testing, just this portion, the polymerase chain reaction. So it is used to detect and measure the quantity of RNA, specifically mRNA, in a sample, like a swab sample from the nose or saliva. It reverse transcribes mRNA into complementary DNA, or cDNA. So cDNA is essentially the DNA version of mRNA and only contains coding sequences of a gene. cDNA is typically only produced in labs when it comes to testing like this, but it can happen occasionally. It is used. The testing itself is used for gene expression analysis, pathogen detection, genetic testing, and other disease research. And more recently, in the past 5 years with COVID-19, we do know it is well known for being used in the detection of SARS-CoV-2 or COVID-19 coronavirus. So here I just included this pathway here of how this works. So first we have our healthcare provider taking a swab sample from the nose, which will then begin our RNA extraction. The RNA or sample is then placed in a viable medium where it is sent off to the lab where we can begin the reverse transcription process. As you can see here, as we talked about earlier, it converts into that cDNA. This is important so we can buffer out any impurities before we input it into the qPCR amplification system. So from here, it is amplified, it is tested, and including different buffers, and this can also be evaluated in different ways through different dye systems. And as this goes through, we go through the analysis, which I will talk about in the next slide, but as we can see here, the analysis is between the fluorescence and the cycle number. So understanding the COVID results from that system we were talking about. So the qPCR software plots fluorescence versus cycle for each well. So it uses fluorescent dyes that bind to the double-stranded DNA to monitor DNA amplification during the PCR reaction. So as we can see here, we have our cycle number and our fluorescence, the background, exponential phase, and the plateau. So as target DNA copies accumulate, fluorescence rises above background noise, as you can see here. So this is what we call the cycle threshold, or CT for short, is where the fluorescence curve crosses the threshold line. So in these tests, it is important to know that the lower the cycle number, it means that it is more of a positive result. So here, the interpretation is if the CT is less than or equal to 35, then that means the likelihood of it being positive for COVID is high. But if it is above 40, we reach this plateau here. There's nothing there. The test is most likely negative. If two or more SARS-CoV-2 targets amplify, it is also positive. So instead of just fluorescence here, we can also target other areas specifically as well. So what does the future of testing hold for PCR and other at-home tests? So PCR testing has rapidly evolved since the pandemic. As you can see, we got at-home testing kits very fast, even vaccines super quickly. But one of the more unique advancements in PCR applications is syndromic testing, which is the ability to test for multiple pathogens at the same time through one test. So instead of having two sticks shoved up your nose, for one for flu and one for COVID, one test can determine it all together. So it's important to, increase our access to these tests for those who are underserved, and also just increasing their availabilities so people can seek treatment faster than they usually would.